So obviously this is biology, which is the study of life. If you did anything with our first lesson in lecture yet, you'll know that all living things are made up of cells, right? Cells are the basic unit of life. So some organisms are just one cell big, like a bacteria. Other organisms like us are trillions of cells big. Those cells all work together to form higher order functions. Cells were first described back in the 1600s, which is pretty crazy. That was a long, long time ago. Robert Hooke looked at a picture like this one down here at the bottom of the screen, cork from a tree, and he found that it looked like it was, when magnified, it looked like it was all these little compartments, and or like a cell, like a jail cell or a room cell, right? And so that's when he coined the term cell. And then people were looking at living things under magnification for many, many hundreds of years. And in the 1850s, the cell theory was established. So let me tell you real quick. Theory is, in science, we use the word theory when we have something that is supported by lots and lots of data and not really refuted. And so it's a way of saying this is, in a sense, fact. We don't really use that word in biology, but saying we have so much data supporting that this is true and no data suggesting that it's not true. So we're going to call it a theory. So we don't throw around the word theory in biology like we kind of do in our everyday lives. We kind of reserve that for something that is very much kind of broad in nature and supported by tons of data. So what does the cell theory state? It states one cell is the basic unit of life. It also states that all living things are made up of one or more cells. And then the last thing it states that is that all cells come from pre-existing cells. So it doesn't address where the first cell came from. It just says once there was a cell, every cell came from the cell before it, and every living thing then was made of one or more cells. And if you get smaller than a cell, then you're not living anymore. Okay? So why do you think it's, and use the chat, why do you think it's useful to study cells? Why do we start here? Why is this where we care? Anybody want to venture a guess? Okay, because they make up life. That's great. So let's say this semester we're going to study single-celled organisms like maybe bacteria or an algae, right? So why would we study these smaller cells? Okay, to help us understand how we function. So if we're made up of single cells just all working together, if we study one cell, maybe we can figure out what's going on in that one cell and apply it to a human cell, for example. Good, to learn more about the world. Excellent. And to see how everything connects. You probably, if you've looked at my first lesson in lecture yet, that's a really big thing for me, is making sure you see that when we're talking about biology, almost everything we learn is related to almost everything else we learn. And so there's this network and maze of connectedness. And when you get to be a good critical thinker, and when you get to be, more of a scientist in the way that you're thinking, you will find that you challenge yourself constantly to make those connections. How does this relate to something else I already know? And how does it relate to the question I'm asking moving forward? So really fun things to ask yourself. Great answers, guys. Good job. All right, so we're going to start with studying cells in here. So one of the first things we need to do is learn about all cells. So if you think about cells or living things that you've seen, just living structures. So think about maybe a gorilla and a bush and an ant and a bacteria, right? They all look so different. Even if you think about in your own body, if you've ever looked at a kind of cells you have in your body, a blood cell looks very different than a nerve cell, right? They look very different. They're different shapes, they're different sizes, and they do different things in our body. So in a way, cells are very diverse. However, if we really look closely, there are some things that are shared by all cells. Now, typically our books, lab manuals, things like that list 4, and I'm going to kind of build on that a little bit because there's more than we can kind of talk about function instead of just structure that all cells share. So one thing that all cells have is they all have a plasma membrane. So that's the border around the outside of the cell. Basically keeps the insides in and the outsides out. And at some point during this semester, in a couple of weeks, we are going to study that membrane. We're going to study the structure, how it allows things to move in and out of the cell when needed. It doesn't allow things to move inside of the cell when they're not needed. So we're going to study that plasma membrane in a lot of details later. But every cell has a plasma membrane. All right. Just an FYI, that is not the same thing as a cell wall. So the cell membrane and the cell wall are different. So just keep that in mind. Not all cells have a cell wall. Also, all cells have DNA, and they store their genetic information in the form of the DNA. So does anybody have, has anybody read lesson 1 for lecture yet? What was my favorite part of chapter 1? Has anybody read it yet? And where I said this is Dr. Bird's favorite? It's okay if you haven't. I know today's the first day of class. I just know that some people have already started. Okay, well, one of the things that's my favorite that we're studying is something that's called central dogma. So what's so cool about cells is, yes, they all have DNA, but what that DNA does is the same in every cell too. And so DNA has information in it that tells the cells to make proteins. And every cell makes those proteins in the same way using the same building blocks. So every cell not just has DNA, but every cell also does the central dogma. So we'll come back to that again a little bit later. All cells also have cytoplasm. That's the fluid that fills up inside the membrane, okay, in which structures will float. So could be DNA floating, could be other structures that are in a cell. And then the last thing that cells have for sure are ribosomes. So ribosomes are small little structures within the cell that build the proteins. So we have the information to build the proteins in the DNA, and then we use that information at the ribosomes to build proteins. All right, and the proteins do everything. They literally are what allow the cell to function and have structure. There's very, very, very few things that the cell is involved with that don't involve a protein in some way. So proteins are things like antibodies and enzymes and structural proteins, all right, transporter proteins. So we'll learn all about DNA and the central dogma, all about proteins, and way more about ribosomes coming up. So don't worry, this is just kind of a preview of what's coming for this semester. But you definitely should know these 4 things. I can guarantee you probably in your post-lab analysis, there's going to be a question that says, what are the 4 things that all cells share, right? And so you're going to have to know those 4 things: plasma membrane, DNA, cytoplasm, and ribosomes. All right, so let's just test ourselves. All right, so here's your first test question for the semester. Draw a cell from the salivary gland of an orca whale. So go ahead, grab a piece of paper, grab a pencil, draw what you think a cell from the salivary gland of an orca whale might look like. Are you guys drawing? Yeah, hopefully. Okay, Sarah's drawing. I'm going to give you another couple seconds. Try to draw the whole cell if you can. Okay, so let's see. What did you start with? Let's see. Sarah, I'm not going to call on you yet. Rosa, are you drawing? What did you draw first? Can you hear me, Rosa? I'm just picking names out. Okay, what did you draw, Rosa? Is that you? Somebody turned their sound on, which is totally cool, but you can hear it. I didn't know. Well, that was Sarah. Okay, Sarah, since you turned your sound on, you tell me what you drew.
What? Oh, it looks, I don't know.
What?
I think I can just show it to you.
Okay, show it to me. I have to, I can't, oh, can you turn your camera on?
This is what it looks like. I don't know what I'm doing.
Can you turn your camera on? At the bottom of the screen, there's like a little video camera. Just click on it.
Okay. It's not letting me.
Okay, well, just describe what you drew. Oh, okay. There she is. I see it. Can you guys see it?
I don't know. I'm doing the circle
it's perfect. She drew a circle. What is that circle that she drew? What does that represent? Good job, Sarah. That represents the plasma membrane, right? That was your plasma membrane. And then she drew white space inside of it. That's your cytoplasm. And she had a couple little structures in there. I'm assuming she was drawing some DNA and some ribosomes. So yes, it's true that probably an orca whale salivary gland cell would be a little bit more complicated than that. However, if you just drew a circle with DNA in it and some ribosomes, like I'm going to show you mine, ready? Like that, you can't be wrong, because that is what all cells like, at least to start with. All right, so let's challenge ourselves with one more question. Ready? Draw a cell from an oak tree root that's deep down in the ground. So a cell that's from a root that's way down deep in the ground. So I have a couple people asking questions. The nucleus. Oh, so yes, it wouldn't have been wrong if you put a nucleus in the salivary gland of an orca whale, because those kinds of cells do have a nucleus. But not all cells have a nucleus, so you have to be careful. So if you didn't know these had a nucleus, then it wouldn't have been right to draw that. But in this case, it was right. Noah says a columnar cell. Yeah, it could have been columnar. That's right. It could have been long. Instead of a circle, maybe you drew like a rectangle. And absolutely, cells have different shapes. They can be columnar like that. Great job, Noah. All right. So anyone, or maybe Noah, you were talking about the oak tree cell, because an oak tree root cell probably is going to be more rectangular in shape. Plant cells tend to be more like that. So now, does everybody feel a little bit more comfortable just drawing a cell from an oak tree root? Absolutely. Draw it like this, right? Show what you have. Show what you know when you can, all right? Don't be afraid to think, well, I don't even know what to do. The last slide just told us that every cell has those 4 things, so always start there, and then you can build upon that. Okay, so let's move on. Now, okay, before I go on, if we were to take all the cells in the whole wide world, they would all have those 4 things. But when we really look closely at all the cells in the world, we could pile them into 2 categories, right? 2 piles, based on the way they're internally organized. Okay, their internal structure determines what pile they're going to fall in. So all cells in the world have those 4 things. And then we have 2 piles, one pile that has this organization and one pile that has that. And those 2 organization structures are called prokaryote and eukaryote. And here's an image of both of them. And you can see that internally they are very different in terms of their structure. This one's very, very busy with lots of compartments that are separate from each other. All right. And the DNA, remember every cell has DNA? The DNA in that cell is packed into this nucleus structure right here. However, if we look at this kind of cell, we don't really see a lot of internal structures. We see the DNA, which is this blue stringy stuff. We see the ribosomes, which are these little dots. And if you look over here, these have little dots over here too. And then this green structure, this green color right here, this is just the cytoplasm. Our cytoplasm in this cell is light blue. And we see the membrane in this picture it's gray, and in this picture over here, it's kind of the inner line on that cell, right? So they very much differ in terms of how they're organized. This one's busy with lots of compartments. This one's far simpler with fewer. In fact, these cells kind of have the 4 things that every cell has and not much else. All right, so we're going to spend a lot of time this semester talking about the differences in prokaryotes and eukaryotes. And in particular, in lab, we're going to be studying single-celled prokaryotes and eukaryotes, and those are the cells you're going to use to base your experiment. All right, so if you don't get it all yet, don't panic, because we're going to spend weeks studying these kinds of cells. This is just a Venn diagram I found online that kind of shows you some similarities and differences. So here in the middle, we see that all of the cells have a plasma membrane. They all have cytoplasm. All the cells go through a process of dividing. Now notice DNA is not on here. And that's because when we compare the DNA in a eukaryote to the DNA in a prokaryote, it's a little bit different. Eukaryotes have linear pieces of DNA, think of straws in a box, whereas prokaryotes have circular pieces of DNA, think of like a rubber band, right? So that's why they're put there. They all have ribosomes, right? And then they don't all, but they can have this structure here. So we'll talk more about what that is later. So some differences between eukaryotes and prokaryotes. First of all, prokaryotes are much smaller, okay? They are between 0.2 and 2 micrometers in their diameter. So let's talk a little bit about what a micrometer is. So if you picture in your head a meter stick, in fact, I should get one and I don't have one at my house. A meter stick is about 3 feet long, okay? If you look at a meter stick, there's 100 centimeters on a meter stick. So all those little numbers on the meter stick, those are centimeters. And then when you look at a centimeter, when you go from 1 to 2, there are, I'm trying to see if I have a ruler at all on here, and I don't. There are 10 little lines. Those are called millimeters. All right. And if you look across the whole meter stick, there would be 1,000 millimeters. If we were to mark out micrometers, which are much, much, much smaller, we would find that there would be 1 million of those little lines. That's a lot. So a cell, a prokaryotic cell, can be 1 millionth of a meter. Eukaryotic cells tend to be a little bit bigger. They're like 10 millionths or 100 millionths of a meter in size. So these are numbers that I expect you to kind of have an idea of, because this is what helps us sometimes determine what kind of cell we're looking at. We can measure the size. If it falls at, let's just say we measure it and it's 75 micrometers, we can feel pretty confident that the cell we're looking at is a eukaryote. However, if the cell is 1.5 micrometers, well, then we might feel very confident that it's a prokaryote. So size difference is really important. I want you to know these ranges. So if I were to give you in a probe analysis, hint, 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 you're looking under the microscope and you see a cell that is about this size, what can you predict? You can make that prediction. Okay? So some of the other things that are different between these, that one of the ones we're going to talk about today, is the presence of structures called organelles. Okay, so organelles are membrane bound, which means they have a membrane around them, the compartments. They're only present in eukaryotes. So you can see over here that prokaryotes don't have any membrane bound organelles. So one of our goals today is to learn a little bit more about the membrane bound organelle. One particular organelle that probably everybody already knows is the nucleus. So we know that some cells have a nucleus and some don't. Okay, so a nucleus is an organelle that holds the DNA in a eukaryotic cell. Prokaryotic cells do not contain a nucleus. Okay, all right. So if we were to just kind of compare the characteristics from prokaryote to eukaryote, if we were to look at their plasma membrane, it's identical. Phospholipid bilayer, we're going to learn way more about it later this semester. if we were to compare their DNA, they both have DNA. Remember, in prokaryotes, it's circular and it's not in some compartment. In eukaryotes, it's linear and it's enclosed within the nucleus. If we were to look at the cytoplasm, you would find in a prokaryote the DNA just free floating in that cytoplasm, as are the ribosomes. Whereas if you look at a eukaryote, you would find ribosomes free floating, and then all those little subcellular compartments, those orbitals, also floating. But you wouldn't find the DNA in touch with the cytoplasm, because the DNA is in what compartment in a eukaryotic cell? Go ahead and type it. Are y'all still there? Can y'all still hear me? Okay, good. Okay, good. You're right. In a eukaryotic cell, that DNA never touches the cytoplasm because it's in the nucleus, which keeps it separate from the cytoplasm. Now, I mentioned the word cell wall before, and we'll talk more about the cell wall next week. First of all, if you're a prokaryote, nearly all prokaryotic cells have a cell wall. All right. However, in a eukaryote, some do and some don't. So plant cells and fungal cells have cell walls, but animal cells don't. So it can't be used as a distinguishing factor always. It depends on what kind of cell parts or cell types you're comparing. And then we're going to talk a little bit about taxonomy. So this little part right here talks about the highest classification of taxonomy. And they're in the two domains, bacteria and archaea, are both prokaryote, whereas there's only one domain for the eukaryotic cells called eukarya. All right, so if we were to look at this, like we said, we started with a big pile of all the cells. All those cells had four things: plasma membrane, DNA, cytoplasm, ribosomes. And then we took all that big pile of cells, and we separated it into two cells. We said this pile is just prokaryotes, and they're simple and they don't have any internal compartments. This pile over here are eukaryotes. They're much more complex and they have compartments, okay, like a nucleus or mitochondria or chloroplast. And then if we say, okay, now we're in this prokaryotic pile, we can separate that prokaryotic pile into two more piles called domains. And we can say one domain is the archaea and a different one is the bacteria. Now, we're not going to talk too much about archaea this semester. When we get to the cell chapter, we'll talk more about it. We are going to talk a lot about bacteria this semester. So next week, our whole lesson is on bacteria. If we go into the eukaryotic pile over here, all of them fit into one category or domain called the eukarya domain. Now, within the eukarya domain, that's where we have, so here's our domains: bacteria, archaea, and then up here, these four little bubbles are the eukarya domain. And we see the plant kingdom, plants are eukaryote. We see the animal kingdom, animals are eukaryote. We see the fungal kingdom, fungi are eukaryote. And then we have this group. It's not really considered a kingdom amongst most biologists these days. Some books call it a kingdom. I teach non-majors biology, and our non-majors book calls it a kingdom. Some books don't. It's a group. And basically, this last group here is anything that's eukaryote that's not a plant, not an animal, and not a fungus. They get put into this group. So it's very diverse. There's a lot of different kinds of cells in there. But what we do know, so if I were to pluck a cell from the fungus category, because it's eukaryote, we know that it has the four things all cells have, and then it's going to have the DNA inside a nucleus.